but really comparatively little impact on impact it has on DNA. Because we know, well, how do we do that? So how do you test that DNA is very stable? What does that mean if it's evolutionarily stable? It means that you have a DNA sequence, you wait 5 million years, and you measure it again, and bloody hell, it's exactly the same. You know, scientists are patient people, but waiting 5 million years to find out is a bit long. So what do you then do? Tell me something, you're the scientist now, and you get $1 million from me, and you prove to me with some good experiments that DNA is stable. What are you going to do? How do you do this?
Just subject it to harsh.
Absolutely. And the nice word is, stress testing. You do awful things to the poor DNA. You put it at -40, and then in 2 seconds you heat it up to boiling it, and you put radiation over it, and you use a sledgehammer, do all kinds of terrible things. And then when you've done that for 2 days, you then say, okay, let's measure the DNA now. And oftentimes, bloody hell, it's still the same. So that's great. So now we believe, if we've seen that maybe evolutionary as well, this might be stable, right here. So that's the idea. So we believe we have sequenced Neanderthals, and the Nisavant was more recently. Old guys, so Roman soldiers we have, they have not changed. It's just recent, right? But this is more the Homo heidelbergensis has been, and we do believe, because we know DNA is so stable, that that is okay. We can learn from it. Yeah? So 10% roughly is Neanderthal in your genomes, and we think that's awful. That's an old sort of human-like species. But Neanderthals have been very intelligent. They think now more intelligent than we. So be proud. Some people have 17% Neanderthals. And they didn't dare to go to parties anymore, because if you tell that story, it's so uncool. But now it becomes cool. They seem to have been clever. Anyway, so here you go. Theoretical image for 1 zettabyte. Maybe we need 10 in 3 to 4 grams of DNA. So 30, 40 grams, 100 grams will do the job. No problem, right? Much better than storing on disks. So all organisms have this fantastic molecule that stores information in a fantastic way. You can copy it with high fidelity, cell division. I told you, roughly 5 to 10 trillion cells you have coming for sure out of a single cell, fertilized egg cell, right? Isn't that amazing? Okay. So evolution is important, right? So now I'm going to give you the game of evolution. What is needed? What components do we need to get evolution? Should I break it for my feelings? I'll do this slide, and then you can contemplate over a coffee what this all might mean. Here, like just 4 things. If you have them, I can tell you, in a biological system you get evolution, but in a computer you can make evolution happen as well. And storytelling is evolving. I'll send you out for a coffee after that story. Okay, so what do you need? You need the tape, the thing that carries information. In our hands, in biological systems, let's call it a template. In a program, in a computer, it could be some string holding information, right? So for us, it's DNA now, and it carries information in a great way, and it's very stable. So that seems like a good piece of to hold information. Now, what do we need to get evolution? This DNA should be copied over. To be inherited, and that's nicely arranged for in most animals and plants, and so you get generations. Parents get children, and these children get children, and it goes on. And at each generation, DNA from, in our way, 2 parents is copied, is recombined and hustled a bit, and then copied into the child, and so on, and that goes on, that process. Interesting, of course, what happens during these copying mechanisms. And then Darwin started thinking hard of it, and they had already an observation. Children are not identical to the parents. We see that. They sometimes look like parents, sometimes they don't look like parents. What has genetics done? Oh, what has the husband done? That might be another question in that sense, but anyway. So that could be a story, but you see that stuff happens. And that means that basically errors are being made. Errors are very important. And the making of errors leads to variation, leads to different things. So sometimes in this copying process, letters that were there on the DNA are copied differently. Normally that's a bad thing happening. Normally if you disrupt something, things break rather than, but sometimes you find as a result, very seldomly, that hey, this is actually a good idea. Errors also in science, they're very important. And then Darwin's gift to the world was in this system. If there is selection, that is sort of giving a system where you can become better. Now, let's take a very simplistic example. You all know an animal called giraffe? Giraffe, right? Long neck, very good. Okay. Now, why would a giraffe have this ridiculous long neck? Looks clumsy, right? It's also beautiful. Anyway. So, a lot of animals eat grass, right? And they eat it, from the bottom, right? From the floor. That's good. So the giraffe doesn't like that so much. It bends this long neck. Giraffe is sort of optimized to eat what leaves? High up! In trees. And why is that a good idea? Because the other animals can't reach so easily there. They don't have that long neck, right? So, for a giraffe, having a gene, I don't even know if that is a gene, it's probably a combination, but let's say there is a single gene and that is sort of length of neck, right? Your coat, the size of your neck. So these trees are growing and evolution demand that trees are getting higher and higher and the giraffe needs to keep up, so you will get selection. And how does this process work? A giraffe mother and father mate, they get a child and fortuitously the gene becomes that for a longer neck. So that giraffe, if that's a bit longer than the other giraffe children, can eat the leaves better. So it becomes bigger and stronger. So there's a better chance of mating again and getting into the next generation. So that's just selection, Darwin's idea. It's a race. Life is a race. And then you see in horses, it's a physical fight. You normally see if two men, identify a lady, they start fighting. I think it's stupid, but if you're a biologist, you say that's good. It's survival of the fittest. Anyway, so this can happen in a physical way in some animals, and in other animals it's very much sort of ritualized. Even, dancing in many cultures was one of the ways to fight as man. So the man that could, dance the coolest, lady, come with me, and stuff like that. So, anyway, behavior and genes, are connected. And for these giraffes, if you can eat more because you have the longest neck and the leaves are so high, that's great, right? So the optimization function is long neck good because the leaves are high. And the other animals, you can sort out what is there to eat low, below, down, below. Yeah, better do that. Anyway, but now you get the cruel thing in evolution. There are ice times, there are warm times. Now suppose that these trees have also their own constraints to fulfill, and now these trees start thinking, being so high up is so cold, I don't know, right? we start shrinking. The trees have a gene and there is some ball game and a fight that the trees get lower, the leaves get lower. So what is the giraffe doing? So that's for centuries or millions of years being optimized. The gene of the long neck was the winner, and all the time you've got longer necks, fantastic. And now it wants, sorry, that's to shorten the neck, the lower the better. So all the fight is different now, and So what do you see? It's normally when we optimize a process, the scoring, the objective function, what is fit is the same, but in evolution, the environment, and therefore these objective functions, what is fitness for the giraffe, might change. Yeah, we've been growing a lot of hairs during the ice times, right? And then it gets hot, and all these poor guys with hairs everywhere. They have to work on it again and lose it, and so on. And then you've just done it, and something else happens. You have to grow like this. So evolution is. So what is important evolutionary? Adapting, of course, adaptation is important, but more importantly, our system should sustain, should have the ability to adapt. We should be able to adapt. It's not just one time adapt to something and that's it, right? No, all the time. So that's very important. Now, this system, if you have these components, the system copying and copying its generations, but with bacteria, they don't mate. They just immediately reproduce without sex. And so you have the error. Error is, if there is no error, there is no life. And selection is if you have a function that one species is fitter in a better position than another, and all the time that seems to be the case. We select at universities. You need to, if you are a poor bachelor student in this country, the discussion in politics now around is you should, of the 60 points that you can get in the first year, you should at least get 42 in this university. Otherwise, we have an advice. We think that it might be a good idea when you try somewhere else. And in earlier years, that was just an advice, but now this advice is compulsory. It's a compulsory study advice. We think it would be a good idea, and then you have to do that. Right? the judge says, I think it will be a good idea when you think about going to prison, right? And the guards take you and whack in prison. That's the idea, right? Okay, anyway. So basically, Darwin also taught us that this, and then I really end up for coffee. There's a conservative process, so I already said that most of the time changing something is not a good idea. It will not add to the fitness of the species or the individual. But every now and then something happens, and if you have enough time to spend, like 4.2 billion years that people think evolution has lasted so far, there is enough time that seldom mutations will overall take place. Okay? So, yeah, I do this. So evolution, I already told you this. Evolution is an optimization process, fitness function, right? Objective function, what is fit? A long neck. How much leaves do you eat when your neck is longer? Sort of fitness, or how strong do you get? Or in the end, you want to measure the chance that you get progeny, children, which is not so easy, but anyway. So this is much akin, you know, what we do in computer science all the time. We have a program, there are 20 parameters, and we would like to tell the users use this setting for this one and that setting for that one. And how do you do this? You do an optimization trick. You let the program run in a whole number of cases and measure you need to measure fitness. And then you say, okay, that setting, you could evolve the parameter values in much the same way as evolution works with DNA. Now, so common applications of minimal cost models, you know, maximal profit. Companies would love to have an objective function to do this. Of course, they're trying all the time. Banks do, doesn't always work. Minimal error, optimal design, optimal management, optimality. You know, optimal means, you know, the best possible, given the situation, the best possible, right? And that's also in evolution. If your neck of the giraffe is long enough, if your neck is longer than the others, you could add another 2 meters, but that won't help you so much unless the leaves are so high that you need more. So enough is a principle in biology, right? Good enough. Sometimes you see a crappy architecture and they say, why the hell would you do this? Well, it works, right? It works well enough. And then the wish to change it and make it better is becoming. So anyway, so an objective function, but you know, as I already said, the objective function changes all the time. Temperature, environments change, and that means it's a cumbersome process, yeah? But evolution is a spatially closed local process. What does that mean? If the child can be different from the parents, but not totally different, right? It's just a few letters different. But over time, if you do it many times, you can become very different, but it's slow, right? So it's a local process. But then, here is some geek speak again. All these creatures compete in space. Sometimes you have a continent called Europe and America, and they were apart once. Animals could just walk and fight together, and then at once you get a rift, an ocean in between. Animals cannot jump over anymore. So you have all of those effects that have a bearing. That's another thing of evolution, compartmentation. I think hopefully Anton mentioned it in cells. Eukaryotic cells have different rooms, if you like. Very important for complexity. And this happens almost spatial-temporal. So time and place is important. One last one. And we really go for coffee, guys. So, stop me when I switch on. We'll go for coffee after this one. So, this is sort of fun. In the 80s, this was quite cool. It was a long time ago, right? So you were not there, most of you at least. But things happen even when you're not there. It's amazing enough. So evolution became really, it's optimization. That's great. So all animals are optimal, right? Life is beautiful. That was the idea. So they said, okay, guys, now it becomes very easy. We have bees, and we know what bees do. The bee dance. They want to locate and point other bees to the food source. They do a wiggly dance, and where 2 parameters are in the corner where they should fly the direction and how far it is. So it's a great system. And they were measuring, okay, how do these? So they made, here is the comb where all the bees were living, and there was a scientist defined food source, there. So they all believed it's optimal. So all the wiggly dance happens, and they all fly up to that food source. What happened if you measure? It was nearly circular. There were also bees that looked at the dance and flew the other way. I think, is that optimal? We don't know. So these biologists were pulling their hair out, bloody hell, we had just so much belief that they would all fly immediately to the food source, and if they do a bit like that, that's okay. But it's all crazy. And then they said, you know what? They started thinking, what can we do? You know what we do? If we average over all those vectors, with a perfect circle of vectors out in all directions, but if you average, you can say, on average, it's pretty, you could say that's in that direction, right? A little bit. So they say, okay, because on average it's a bit more in this direction, and there we call it optimal. Think about for a moment what does it mean when on average you're optimal. What's the definition of optimal? The best given the condition. When you have an average, normal distribution, an average means it's average, right? So it means some stuff is better, longer, taller, better, stronger, and less. So it means you have species that are better than optimal. Do you get it? Think about it over coffee. Right. Can we be back? Sorry. It's going to make a story. If you take 10 minutes, is that okay? You have a right for 15, I know, and those who feel like taking the 15, I won't say anything against you. You might have a bit of a problem if you want to find your seat, someone in the middle later on. Yeah. Okay? There is a coffee machine over there and drinks. Yeah.
I have a few questions. I just wish I could, but that's okay.
I'll take it. So if you don't have time, I will let you
something about one of the requirements being copied, but in a parallel world, perhaps copying the context. If there was any dynamic, if they could change, would that? No, I understand copying is a good idea, but is it really a requirement? Because it has itself, and that can change. It's during its lifetime. Exactly.
Now you have an enzyme. That sort of stuff.
Yeah, something about unraveling of the nucleus stone.
Yeah.
I was wondering, so obviously the polymerase, sorry. Is it, so the polymerase needs to be able to go through the thing, but it cannot do that while it's around the history.
Yeah, the protein. There can be even more, so the view was that there can be more polymerases copying in, start sliding, so you can do that more than the trains. One thing is the same, so this was halfway
No, I understand. Okay, but
what happens here? I think DNA is not going to do things.
Yeah, I know there is some histone modification so that it loosens it up a little bit.
And helicase, there are, I don't know if I have the slide, but there are, if you copy DNA, so cell division, double should be taken apart, one cell, and then the other strain should be made, so it's like it has to be ripped out, it's protein, and then there is the calcium element that keeps them apart, so you put the thing, okay, because DNA wants to go back, so it keeps it open, and then you enter a special protein to close the stuff again.
But it is simply a huge mechanism. When you eventually have it, you have all these apparatuses around this string of, it's amazing.
But if you think about it in the information sense, transcription factor what the DNA can tell the system how to act. And that's amazing.
But also like the loop between transcription factors being proteins and being encoded.
Yeah.
Amazing. Okay, good.
Yeah.
Good. I'm gonna have some fun. Sure.
Yeah, same for me. En zo. Ja. Maar belangrijk is om te onthouden. Kijk, de DNA molecuul is van dat motief hier en dat hier. En dat was Darwin en dat zie je dat is van de Duitse plaatsvinden. Ja. Het maakt het overzichtelijk. Ja. Weet je, de mRNA gaat naar het ribosoom en dan wordt het getranslateerd in een eiwit. En dan wordt het doorgetrokken. Dat hoeven wij ook niet druk. Dat is ook wel niet weten, maar het is nog belangrijker dus om bij je studeren te verstaan. Ja, dat is een boeken van de maar omdat het allemaal lastig is, ga je dat gewoon weten of niet. Als wij dan helemaal klaar en dan weten we dat dat oefenen we ook daarmee. Ja. Dus als je nou een beetje worstelt, dan is het helemaal goed. Het klinkt een beetje klaar, maar het is en als de verwarring blijft, dan kan je niet blijven. Dan ga je eerst even naar alles. En dan doen we duidelijk aan. Ja? Right?
Just check it out.
Okay, guys. Ladies and gentlemen, dear travelers, as they say in the Dutch railway system. Because saying ladies and gentlemen is not good enough nowadays. Yeah, I could say ladies and gentlemen and all in between, but that sounds also a bit, I don't know.
What's in between?
Transgenders, you name it, yeah, whatever. Okay, there is one principle with your eyesight, so I would like to start quickly because there are quite a few slides coming up. There is a very nice physical principle which allows you to go into a cinema and watch a movie that is something called flicker fusion frequency. Flicker fusion frequency. And that means that you look at pictures, you see them, you turn pages, but if you start turning pages at a frequency of 25, 26 hertz, so 25, 26 times a second, the eye is not able anymore to see the different pictures when you see a moving image. Right? That's a film. A film is nothing else, a movie, 25 pictures per second generated, right? So we have 25. You know a bee? Homology on the nose. Flicker fusion frequency is roughly 300 hertz. So some animals do that much better than we. Some people are cross-eyed, by the way. Right? They look, your eyesight should. In the corners of your eye, your flicker fusion frequency is a bit higher. 60 hertz. And that means that in the old times, cross-eyed people sometimes have trouble watching television because one eye would be such that they would see the flicker. So one eye sees something move and the other. Anyway, and why am I saying this? Who can guess? I have so many slides that if I go on talking like this, I have to play them past you at the frequency over 25 per second. And that would mean you see a movie, but it would be not a very consistent movie, I guess. I don't know. We could try that once. Just play them as a movie. Anyway, we're all about evolution. This you should know by heart. From now on, I start talking about, you see me about 4 o'clock moments, meaning 4 o'clock at night. I come in your room, you're nicely sleeping, you thought. I wake you up, and I ask you, what is homology? What is orthology? What is paralogy? You just tell me the stuff, and I'm out. No more trouble, okay? So that sort of thing. So that means, you should really know this because this is one of the very principles of evolution and biology. If you will talk to a biologist and you don't know this, the guy will turn his or her back to you. And that's what you don't want to have happening, right? So what is happening? So I give you straight the definition of homology. It's a very simple one. You have a gene. Simple man's picture. You have a cow. There is an ancestral cow. I don't know how much it looks like a modern cow, but there was an animal once before that was cow-like. So it was eating the grass and so on, not as nicely as the trees as the giraffe, but anyway, it would find food sources on the ground. But then, and it was living on this continent, America, Europe. It was one big thing, right? And then there was the divide of the continents, and some cows were in the European part, and some of the cows who were sort of very similar were in the American part, and then the environment started changing, cold in the one, warm in the other, humid here, dry there, all kinds of things, and that means the DNA of the cow started adapting. And after a few million years, we were looking at these cows, all originating from this flock of ancestral cows, and they were very different. They become different. Right? That is what you very often have in biology. You look at something, and homology is a principle that ties it together. It says, okay, we have a gene here in this species and a gene there in this species, but hey, maybe there's a reason to believe that it originated in a single thing, okay? When that happens, it's homology. We call that homologous. Having a common origin. Now, a lot of biologists, and even mathematicians sometimes, they talk about this is a little bit homologous, right? This is more homologous than that. That is in principle a nonsense. It's about saying this lady is a little bit pregnant. It doesn't work. It's a binary thing happening, right? You are or you're not. So also here, binary, you're homologous, meaning you have a common ancestor or you do not have a common ancestor. No, you're not homologous. That's what it is. You can be a little bit similar and a bit more similar. That's the confusion normally, right? And then people call that homology. It's not. Anyway, that's one thing. Now, there are two ways of having a common origin. In biology, this is very interesting. If you have a cow and a horse, they might have a common origin. And then you call the gene where this happens orthology. Sorry, difficult word, but it just means this is the situation. You have a cow gene and the cow genome, the horse genome, and this is the corresponding gene, which was the same gene in the ancestral. They both derive from one thing. So these are homologous genes. And the whole genomes, of course, are homologous because the ancestry includes the whole of the genomes. So that's one way. And biologists sometimes call it copying, but it's copying from the common origin. Okay? Do this do these genes need to be the same? We know one thing for sure. In the beginning they were the same. But because the one was in America and the other in other dips, so they start becoming, according to Darwin's rules, they can become very different. And you call that process divergent evolution. Yeah? You start common origin and then you start becoming different. You diverge. Is that a question?
Convergent evolution.
You have that, but I won't talk about it today. Happens less. Can be very important sometimes, but you know the overwhelming evolutionary processes are divergent. But good point anyway. So this is corresponding, and then what can happen, and this alludes to your question in the break. Right. I'll get more on that. Is that you can have within a single genome. Now one of the things again is I don't know if this is a deception, but it's interesting. We genomically, according to our DNA, we are complete stutterers. More than half of our DNA has bits that are just repeats of bits that were there before. A gene that has been copied. There are whole mechanisms that happen that copy stuff over, so genes land somewhere else in your genome. It has to do with this word again, read it and see, okay? So anyway, this is what then happens. You have a gene in your genome, and somehow by a mechanism, that gene is copied over 100% to another place on the genome. And that means if that bit would encode for a protein, chances are if the whole gene is just transplanted somewhere else, or copied somewhere else, that sort of gene now also starts producing that same protein, right? Or the system with the polymerases and the transcription factor binding sites will sort of facilitate the formation, also using this gene to make the same protein. Now there are two places on the DNA that makes the same protein, right? This is called, for a short moment, a paralog. So this is between species, between genomes of different species, and this is within a genome. Paralogy. Orthology, paralogy. These are two orthologous genes, and these are paralogous genes. And now the situation can even be more crazy. Here is a sort of famous textbook picture where there was an early globin gene, globin hemoglobin, oxygen transporting protein. This was an early gene. Then a gene duplication took place. So here, what is this? This is paralogy here. So you got the first thing happening is that this was in two genes, the same gene. We need to give them both names. So this one we call alpha, and that one we call beta. So then we're here. And now something happens called speciation. So this gene, and then this was Africa, and this became America, and this became Asia or something. A mouse, a chick, and three different species. And these are, what is this? Speciation, formation of species. So this is paralogous genes, alpha, beta are paralogous. These frog, the alpha gene in chick, frog, and mouse are orthologous genes, right? Now, you can have orthology following paralogy, but it maybe could also be the other way around. Now, look at this tree. How likely would it have been, now you're an evolutionary biologist for a moment, right? How likely is it that first you would have had speciation and then duplication? How would the tree have looked? What would you get if you get speciation first? Then this bit is here. You get 3 times an early globin gene in 3 different species. And then what? Each of those 3 independently need to then say, you know what, I do a copying and I go in alpha beta. Now, not knowing anything, what would you guess? And the textbook guessed this situation. Right? So very often, we don't know. You know what the bummer is in evolutionary times and common origins? They're not there anymore, they became extinct. It was millions of years ago, right? And we have to start reasoning. That's evolutionary biology. Anyway, orthology, paralogy, remember it. And binary homology. Yeah? Okay, now fill that in a little bit more. Here we go. Evolution, the timeline runs like this. We have a gene, it's a very important gene. So Darwin tells us, oh, selection pressure is up. Meaning, if you change it, you're dead. This gene is very important for getting children. If you mutate it away, ooh, very sensitive. Don't do it, right? And if you become sterile, because there is a sterile gene, if that clicks in, you become sterile, that's not good for getting children. So, here is a very important gene. Then, at a given moment in time, there is this gene duplication. You get 2 paralogous genes. Who can understand that now at once the selection pressure goes down a lot? I give you the buzzword. Redundancy. What happens? At this moment in time, you get 2 times the gene encoding that very important protein. So now you start mutating one of those genes away. You start changing it. Is that very bad? No, you still have the other copy that does it. And whether it's the original copy or the original or the copy, that doesn't matter. But that's what happens. So, but then, suppose this happens. So this, say the B copy, the beta copy is mutating away. In the beginning it's doing roughly what the other guy is doing, and then later on it's doing something else, something completely different. What then happens with the selection pressure? The gene is still very important, so you restore the selection, it gets maybe back to the original. But what have we as a result, because of this copying? A new gene, a new protein. Maybe that's a great thing. So, redundancy is the playground duplication of getting new functionality. That's evolution. Okay? And then, biologists beat each other to death very easily in terminology and so on. So you have sub-functionalization, that is sort of, you have an old task, a protein, and it sort of splits up. You get a duplication, but then they still add together, and one is, the process is now subdivided. You call that sub-functionalization. And getting something entirely new, this copy mutates away to something entirely different. It's called neo-functionalization. There are mixtures, of course, but sometimes biologists, with all due respect to the biologists that have been trained like this, are trained on these principles and terminology, and they find it important. Sometimes you think, yeah, but you can say that, but we can't observe it, so why fight about it? Anyway, so this is all what happens if you have genetics and genomics and sexuality. Now think about this scenario. You are in New York in a huge skyscraper. You have to go 60, we don't have buildings like that here, but you have to go up 100 floors. You stand in the elevator. A lot of people stand next to you in that elevator. You get out of that elevator 40 floors higher and you have genetically changed. The question is, what happened? And this is not a realistic story for people, for mankind, but for bacteria it is. Bacteria do this all the time. They don't, they are also in elevators, by the way, all the time. But anyway, they have 3 different mechanisms: transformation, conjugation, and transduction. You don't need to, this is not a 4 o'clock moment, this is just to illustrate the point. You don't need to memorize this, to just show it, that you see how diverse biology is. So they can exchange their genetic material by just lining up, being next to one another, building a little bridge, 3 different principles of, Do you know what the big scare for healthcare on this planet is now? Bacterial resistance against antibiotics. This is because of this. One of these bugs gets resistant, and it's in the elevator, and it gives that resistant gene to all the others. Now they become resistant, and this can just happen any moment. Yeah? So that is, so if you have one resistant bacteria in your system, in no time, others will follow. And that's not good, because then you need to kill those, and then you need to develop something else, and that's a real problem. Okay, so quickly a few slides. This will also be in your biology class. So you have mutations. Basically, you have different mutations. If a letter changes on DNA or protein, people talk about a point mutation. You could also have that the whole stretch of DNA is taken out or put in, insertion, deletion. We'll talk about this at great length. But for now, a single letter changing mutation, we'll call it a point mutation. Here are different. You have AG is called purines, CET pyrimidines. This is a bigger structure than that. You can look it up in the textbook. So if you have a mutation, that a transition means you go from A to G or vice versa, or from C to T or CU and vice versa. And if you go from A to C, that's called a transversion, because you change the purine into pyrimidine for the other way. This is maybe a more important slide. Biologists like this terminology. I told you, you can change a letter in the codon at DNA. Sometimes your amino acid will change, but if you do it at the 3rd position in the codon, remember, oftentimes it's a silent mutation or a synonymous mutation, meaning the amino acid will not change. And if you do another change, the 1st or the 2nd position, normally you get another amino acid as a result, and that is called a non-synonymous mutation. Makes sense, right? Now, if you talk about something that changes a letter, an amino acid, there are 2 types of change. You change an amino acid in another amino acid, or you change an amino acid in this funny amino acid that's not an amino acid. You have 3 kinds of those to stop. could happen. And then you call that a nonsense mutation. Right? So you have missense, one amino acid to another, and nonsense meaning an amino acid being mutated into stop codon. Okay? Now, and again, now you have the whole story here, transcription from DNA to RNA. I showed you the process a little bit, and then you get the translation at the ribosome from these codons, 3-letter blocks, into a protein sequence via the encoding, and this is called the universal coding table, meaning in all species they do it like this, and we were very sure about this as biologists. We're all happy, at least there is something universal, great. And then you find out, bloody hell, a couple of bacteria, they just do it a little bit different. They have another encoding. So that can even happen. So you have very basic principles sometimes that you think should be universal, and then you find out even that mutates away. Anyway, yeah.
But do they have the same proteins?
They have the same proteins, it's always 20 units. So they just say, okay, if you do that, now you become that, and a little bit. Not crazy, but it's different. Okay? I'm telling you not to confuse you a little bit. So I like confusion. This is a strategy that's also in politics playing a big role, so in science we don't use it. If you feel totally confused, I say, I reached my goal. Okay. And this sounds like a joke, but in the beginning times you'll feel like this. And I told already a few of you, trying to be serious, this is maybe even a good situation. That you are bewildered by the complexity and feel like, I really need to learn this. And then we start filling that in if you develop a design. Anyway, how are we going to work as bioinformaticians in this? Basically, there is one overriding principle, just one. You can now already memorize it, it's done. Homology is your big word for the next 2 months in this course. Homology. Because that helps us. And it's, what's the beauty if you're a bioinformatician? You can become very lazy. You like that? No big experiments, no lab work, no typing up programs, code. No, there's just stuff available. You press a button and you can learn about the system and about, for example, find the function of a protein. I told you, proteins are the doers in the body. Many diseases are connected to proteins. I mentioned Alzheimer. So finding out what proteins do, how they fold, what their structure will be, what they, as a result, will have as function, is very important. We know a lot by measuring, but an awful lot we do not know. Typically, if we find a new bug nowadays, a new species, a few years ago I said roughly half the genes we can find what they do, and the other half, now that's a bit better, but I think 70%, so 30% will be a question mark. That's too much to understand the system. You go to a car mechanic and say, I've got a problem in the car, and this person tells you, sir, I understand 70% of the system. They're going to say, I visit another one. Have a nice day. So that's the situation. So we need to do better. That's why you are sitting in the class here. You will make these things better. Okay? So you'll know. So anyway, in this course we start